Hello, everyone. Good morning. Thanks for listening to my Sloss Talk this morning. I'm Matt Selensky. I'm a fourth-year PhD candidate in Dr. Maggie Osborne's lab, and my research focus is cave microbial ecology. And so I have a bunch of different field sites that I have samples for. They represent lava tubes, continental karst caves, as well as submerged karst caves in the Yucatan. And I basically have all these samples and I'm doing like DNA extractions and lipid extractions, but kind of the uniting question that is guiding me through my research is this one up here, which is what is the distribution and activity of chemolithoautotrophs in the shallow subsurface? And those are those microbes that fix inorganic carbon into biomass using inorganic energy sources other than sunlight. So what I kind of want to talk about today is my work in the lava caves at lava beds, because we just published a paper on this summer. So Lava Beds National Monument is in Northern California, and what we did was we extracted membrane fatty acids from microbial communities living there. And so we looked at the structures of the fatty acid moieties here, and then we analyzed their stable carbon isotope compositions to try to track their carbon sources, whether they're getting energy from photosynthetically derived carbon, so they're heterotrophs and not chemolithoautotrophs, or if they're gaining a lot of their carbon from carbon fixation in the cave itself. And so we kind of had some surprising results. What we found was that we found this really weird fatty acid called 1014-dimethyl pentadecanoate, which is right here. And its source is actinobacteria, which is this really metabolically flexible phylum of bacteria. And when we looked at their carbon isotope compositions, we found that the delta C13 values were like -43 per mil. And when we compared that to other carbon reservoirs in the system, we did a simple mass balance calculation to try to understand, are they getting their carbon from chemolithoautotrophy or surface photosynthesis? And so the way to read this plot is that things on this end are more chemolithoautotrophic and things on this end are more influenced by surface photosynthesis. And what we found was that in the biofilms, in these kind of blue humps here, the lipids found in those biofilms, including this really weird one from actinobacteria, are super influenced by chemolithoautotrophy. And so we kind of had evidence that we had this widespread carbon fixation in these caves. And if you're interested, I invite you to read about it in our recent publication in JGR Biogeosciences. Kind of coming soon, I'm hoping next time I will talk about these other cave systems that we had. I'm kind of shifting gears now and doing a lot more DNA sequencing work, looking at kind of the core community throughout the Yucatan carbonate aquifer and microbes living in Mammoth Cave and Hawaii. I just don't have a pretty picture for that one yet. Some other 2021 happenings that I kind of want to mention. Since February, I've been a student research consultant for NUIT, which has been really fun, helping people on board on Quest and use that supercomputer. I also was able to do some Mammoth Cave field work with Maggie and undergrad Zoe that we work with. And I've also done virtual outreach at Blanco Elementary School in the Hill Country of Texas. So thank you.